it is a great privilege and pleasure for me to introduce my colleague, Andy Cohen, as today's speaker for the colloquium. Andy is a distinguished professor of geosciences at the University of Arizona, and he's our associate department head as well. Andy also holds a joint professorship at EEB at the University of Arizona at the Department for Ecology and Evolutionary Biology. And originally, Andy hails from the northeastern US, and he got his bachelor's in, I think you double majored, Andy, in geology and biology at Middlebury College. And then at the time, there was a lot of kind of revolutionary things going on in the world of paleontology. And I guess Andy was kind of inspired by ongoing debates about whether the fossil record is actually legitimate or whether taphonomy and the rock record owns the fossil record. So I guess Andy was inspired to basically go out west and work with Jim Valentine, who was involved in these debates with Sepkoski and David Ropp and others. And so Andy went to UC Davis to pursue his PhD with Jim Valentine. But unfortunately for Andy, or maybe fortunately, Jim Valentine basically took a job at UC Berkeley as Andy arrived there. So even though Andy hasn't, he went to Santa Barbara, right? So, but even though Andy's not necessarily told me, I think I've reconstructed that Andy probably did a lot of his PhD just purely by himself. Although he did have a lot of help from people like Leo Laporte and Kay Berensmeyer, who's currently the curator of vertebrate paleontology at the Smithsonian. But Andy did a lot of really seminal work during his PhD at UC Davis. And in particular, sort of famously, Andy worked on several aspects of the paleolimnology and the geology of Lake Turkana in East Africa. And apart from sort of characterizing the basin history and stratigraphy and sedimentology, Andy also extensively worked on invertebrate paleontology and the paleoecology of ostracods in Lake Turkana. And his work eventually helped characterize sort of the Neogene paleoenvironmental context of East Africa, which of course is very important for our own human origin story and sort of the important fossil record that exists there and contextualizing the debates that are going on there. And this kind of gives you an insight into Andy's trajectory and career. He's a very extremely broad thinker, and his earth scientific interests touch upon a vast array of subdisciplines and fields, but he's got a whole other limb in terms of his biological interests and sort of paleontological interests as well that touch upon several subfields. So I'm not going to bore you with examples of what Andy's done, because you can go look up his scholar or CV. He's got a very storied career touching upon many different aspects. And building on that, he has several awards as well. Again, there's too many to name, so I'm not going to name all of them, but I will say that Andy received the GSA Limnology Division Israel C. Russell Award, and he also won the International Limnogeology Association Wilmot Bradley Medal, and he's also a fellow of the American Geophysical Union, as well as a fellow of the Geological Society of America. I'm not going to go on too much longer, even though I could. And Andy, I just also want to say that it's been a pleasure being welcomed into co-teaching with Andy, and I'm sure many other early career faculty have had that experience as well. So without further ado, Andy, take it away. And thanks for agreeing to do the colloquium.
Well, thank you, Cal. That was very kind of you, and I appreciate everybody being here today. Hopefully we won't have too many more of these Zoom sessions. I know we're all getting tired of this, but I was excited when Cal asked me to give a talk, and originally I thought I would talk about our hominid site drilling project, but then I thought I want to talk about things that I'm sort of cycling back to after a number of years away from them with an upcoming project that we're hoping to get off the ground. So all of this is around this theme that is shown in the title here with Lake Malawi in the background. And what I'm going to tell you about is not by any means only my own work. It's the result of collaborations when you're involved in scientific drilling you can't help but collaborate with a lot of people, and especially the last 10 years with being director of the Hominid Sites and Paleo Lakes Drilling Project. I've had lots and lots of collaborators, as well as many great students, some of whom are listed here, who have contributed to what I'm going to tell you about today. So I want to start off by just going back in time 160 years to a probably what was then a minor story in science and biology, although there was a lot of interest at the time among European explorers who were returning from expeditions to the Great Lakes of Africa, and bring back all kinds of very interesting specimens. And I'm showing you some here that came from Lake Tanganyika. And I just want to read this at the bottom. Mr. Edgar Smith described 2 new species of shell from Lake Tanganyika. These forms are without doubt generically identical with Pergulifera humerosa, only known to occur in Stratolaramie group, extensive brackish water formation in Western North America, transitional between Mesozoic and Cenozoic series. And actually, when you look at these, here's a couple of Tanganyika shells, and there's some other ones illustrated here on the left, but these 2 living ones, and you can see the superficial similarity between this guy that's labeled number 1, which is Paramelania, the genus Paramelania. These things are entirely endemic to Lake Tanganyika, and it's just a matter of convergence on these marine forms that led people at that time to come to this conclusion. But they were really onto something, and the thinking about this was stimulated even more in the years to come in the late 19th century. I'm going to show you a video here. This is from an ROV dive, and I'm going to show you a couple of videos actually in this talk, but I want you to see what's floating in the water here. And I'll turn the sound off here. And you can hopefully see thousands and thousands of jellyfish. And if you've ever gone swimming in a lake, probably you've never seen a jellyfish in a lake. And that's for good reason, because there are only a few places in the world where there are jellyfish in lakes, and Tanganyika is one of them. And that further stimulated this idea that this is one weird lake with some kind of the ideas that were floating around from that earlier picture I showed you. Maybe this was some relic Jurassic seaway, or people didn't know what it was, but why were all these weird animals there? And so I want to use that as a framework for this talk. And I want to discuss a number of points. One is why we go to the trouble to collect these long, and by long I mean ranging from tens of thousands to millions of year-long records from the African Great Lakes. And secondly, I want to try to answer some questions, some fundamental questions in ecology and evolutionary biology that these drill core records can address. And I think we have some good systems in these lakes to do that. I'm going to tell you about some case studies from our past work and our current work at Lake Malawi and Lake Tanganyika, the 2 largest African Rift lakes, and also our plans for drilling Lake Tanganyika going forward, because we've only collected cores going back a mere 100,000 years in that lake from piston coring. So before I dive into that, though, I want to make it very clear that I also see these long records as being essential for putting the modern environmental changes that lakes are experiencing in context. So I like to think of this as kind of the past as the key to the future. And specifically, these 2 cartoons that are here on the left side of a cooler lake, the world of Lake Malawi and Lake Tanganyika, say in the late 19th century, versus the conditions that occur today as the lake is warming. And in the process of becoming warmer at the surface, it's cutting down on the mixing of the water column. These lakes don't overturn the way the temperate lakes do in the fall and spring. And that's locking up nutrients in deep water, preventing them from coming up to the surface, and having a huge effect on things like declining primary production. So if you can't bring nutrients to the surface, you can't bring fish food up to the surface, and that reduces the productivity for the enormous fisheries that exist on some of these lakes. So there's a very much a importance of understanding the past as a means for forecasting what might happen in the future when we look at past ecological states. So why we drill these lakes is really to get at these long continuous records and provide us with what I like to think of as multiple historical experiments. What's happened in the past under conditions that are similar or different from today. And these core records are great because not only do they give us a long record, but most of these rift lakes are very steep-sided. There's not a lot of outcrop exposure in many of them. And these high-resolution records, like this varved, annually laminated piece of core from Tanganyika that you can see here in the middle, give us the potential for looking at very long records going back in time, even millions of years. And all of these lakes, I've been involved in drilling these three on the bottom here, Lake Malawi, Great Salt Lake, and Lake Magadi. I was not involved in the Lake Ulkrid drilling project, but I just wanted you to see, to get an idea of what we in our community of the Continental Scientific Drilling community have been able to accomplish, drilling in that case down almost 700 meters below the sediment water interface. So potentially very long records that you can obtain from these places. And furthermore, the fossil record in some of these, especially these stratified lakes, can be phenomenal. You have excellent preservation, and I'm showing you here the head of an insect. You can see the eyes here. And this is so well preserved that the musculature and integument is preserved here. Here's a shell. These are both from Malawi. And the original shell color is preserved in this specimen. Both of these about 100,000 years old. So there's a lot of potential, not only for looking at body fossils, but also molecular fossils, particularly for the potential for doing ancient DNA work on understanding the phylogeny of these based on this exquisite preservation. So to date, scientific drilling has been done on a lot of ancient lakes. Here's just a map of the world showing the locations and the dates where these were drilled. And these lakes, in some cases, date back, like this one here in northern Siberia, Elgigitkin, dates back over 3 million years. Some of these are shorter, not snapshots, but cover shorter intervals. But we have succeeded around the world now in collecting records in a lot of different places, in a lot of these ancient lakes, particularly in these tectonic basins. El Gigitian happens to be a meteorite impact, but other ones like the African Rift Lakes provide us with the potential for going back even further in time. And I'm going to come back to that at the end of the talk. Now, I alluded to this at the beginning, that these African Great Lakes are reservoirs of not only extremely weird organisms and things that often are convergent on marine organisms, but also extremely diverse communities. And I'm just showing you here with these histograms the diversity at the species and genus level of a bunch of these lakes, especially in the Western Rift Valley. And Tanganyika and Malawi definitely stand out. These are log scales, notice. So not only in the total numbers of species that are present here, which is often for certain groups like the cichlid fish or some of these mollusks numbering in the hundreds, but when you look at the genus level, you can see that Tanganyika stands out because not only are there are a lot of species, but they're really different. They're really weird. And in many cases, they do show these remarkable convergences on things that we find in the ocean. So I like to think of these lakes as maybe sort of having the ecological characteristics of an incipient ocean basin. And it may not come as a surprise that the oldest lake in the world, Lake Baikal, also has a fauna that is very diverse and has the same kind of characteristic to its diversity. So the diversity occurs among fish, but also a lot of different invertebrates. snails, crustaceans such as the ostracods that I work on, and many other types of organisms. And I want to again show you a little bit of video just to give you a sense of what it's like swimming in a fishbowl. This is ROV footage from Tanganyika from a dive that we did many years ago now. I'm embarrassed to say how many years ago this was, which accounts for the not super high quality of the video. But you'll see in a minute why those of us who work here get so excited about returning to this place every year. So let's take a look. I'm going to turn off the sound, and you'll see here in a moment just what an incredible fishbowl this is, with many, many species of fish. And this would also be true. You can see some of the snails on the rocks there, but many varieties of fish, very colorful, things that you would otherwise kind of squint and say, I must be on a coral reef. And indeed, there are stromatolitic reefs, stromatolipalgal reefs in this lake as well. So a lot of habitats, again, convergent on what you'd see in the ocean. Just to put this in context, at this one small area of the lake, there in a couple of days of surveying this was a study looking at sedimentation impacts, we recorded twice as many species of fish in just about, let's say, 10 or 11 50-meter transects as exist in the entire state of Arizona in freshwater. So that's just an indication of the diversity here that we're dealing with here. That diversity is accumulated through both great age, and some of these lakes are, like Tanganyika, are really old. We estimated many years ago that Tanganyika is probably on the order of 9 to 12 million years, and that number has held up over the years. And in fact, the molecular phylogenetic molecular clock calibrated independently from the age of the lake gives a similar kind of age. So on this cladogram that you can see here, I've got the cichlid fish. Don't worry about the names, there's no test. But I want you to just notice the difference in how the diversity is deployed. And for those of you who haven't looked at one of these kind of diagrams in a long time, the present is here over on the right, and just some pictures of some of the iconic varieties of these fish that are present here. And you can see that the divergence times in Tanganyika go way, way back. So even though the numbers of species actually are less than what we find in Malawi and Victoria, the divergences are very deep. And it's that deep time, long time period for differences to accumulate that accounts for all that generic and even family level divergences. So in Tanganyika, we actually have endemic subfamilies of animals, and that's really extraordinary. So what are the big questions that we think we can ask from long records from drill cores? Well, and I'm going to give you some examples of these. We can look at how ecosystems respond to extreme perturbations. These lakes have been around for a long time, and I'm going to show you some examples from Lake Malawi and what happened when it almost dried out. We can look at the tempo of ecosystem change through long-term shifts in climate and tectonics. Remember, these are rift lakes, so they're affected by a variety of different kinds of forcings. And we can constrain that phylogenetic or evolutionary history of these different species groups, what we call species flocks, and really use this as a model for understanding the time and the mode that these large adaptive radiations have occurred. Malawi and Tanganyika are textbooks examples of adaptive radiations. You'll find discussions of them in almost any evolutionary biology textbook, but it's almost always from the perspective of looking at molecular genetics of the modern species. Very little fossil data because we don't have the long records for the most part. Let me start by talking about Malawi. This is a very deep, very big lake seen in the background here. It's probably about 5 million years old. And as I mentioned earlier, it's a stratified lake, so it doesn't overturn. animal life is restricted to the upper part of the water column, and below a couple hundred meters down, all the way down to the 700-meter bottom, it's anoxic, and life is restricted to microbial organisms. Lake Malawi is down here at the southern end of the western branch of the African Rift for orientation. We've known that Malawi was not only an old lake, but also that Malawi had dried up at times in the past. We've known this for over 30 years from work by my colleague, Chris Schultz, and his PhD supervisor, Bruce Rosendahl, from a big seismic reflection project called Project Probe on the African Rift Lakes back in the 1980s. This was the cover of Geology on one of the issues in 1988. And you can see this beautiful image with this big unconformity here that represents a major drying episode of the lake. So we knew that the lake had dried up in the past. And here's a much more recent multifold line that you can see much more clearly evidence from these onlapping reflectors and this ancient, interestingly, this ancient buried paleodelta here sitting about 300 meters below the lake surface today. We knew that the lake had bounced up and down in the past. What we didn't know and couldn't know from the seismic alone is when this happened. We needed dated cores to do that. So in 2005, after many years of knocking on doors at NSF and trying to convince people that we weren't crazy, my colleagues, Chris Schultz, who's not in this picture, Tom Johnson here, recently retired from the University of Minnesota Duluth, Mike Talbot, and others, John King from University of Rhode Island and others, finally got funded to do this drilling project at Lake Malawi. And what you're seeing here is the drill barge. It's also in my background. So when we started this project, we didn't have a JOIDES resolution to go out on. We had nothing. And what we had to do is find a barge of convenience that was operating on the lake. This one here, the MV Vifia. And this is what it looked like in 2004 before we hired a naval architect to build that drill ship. And interestingly enough, so these are these four 250 horsepower engines that you see in the corners of the barge here. These are the dynamic positioning system that keeps the barge stationary. And then we drilled down to the lake floor with this big drilling system. And here we are up the top looking down. You can see there's not a square inch of space on this deck. We had to set up the core extraction rack on top of this pipe. So everything was incredibly dangerous and tons of fun, but it was, looking back on it, kind of a crazy project. So we ended up getting some great cores after a lot of initial disasters. And here again, the barge, and here you can see the penetration of our longest core, which went down about almost 400 meters below the sediment water interface in this now deepest part of the lake. And I want to just show you, because I'm a paleolimnology nerd, so I can't avoid showing cores. But even very shortly after we started pulling these cores out and opening them up, we realized that the seismic data that we saw from Chris's work many years before was borne out by the nature of the cores. You can see this clearly without knowing much about sedimentology at all, that we saw alternations between the kinds of deposits that are being laid down in the lake today. These very finely laminated diatom oozes, no calcium carbonate. These are typical, very organic rich, typical of what you'd find in a deep water stratified lake. And then prior to about 80,000 years ago, as we learned later once we started getting dates on these things, we saw things like these calcium carbonate nodules, evidence for paleosols, a lot of evidence for even desiccation at times, indicating that this drill site, which is today in almost 600 meters of water, had completely dried up, and the lake was reduced to kind of a mud puddle. So my role in this, and my students who work with me, my students and postdocs who work with me, we're looking at both the aquatic ecosystems. So we had a fantastic ostracod record that I worked on with Meg Blom, diatom record that my postdoc Jeff Stone worked on, and then insect and mollusk records I already noted a second ago. And then Sarah Ivory did the pollen work, and Ed Yost the phytolith work. And so I'll show you a little bit of that data. First, of course, I got to show you the ostracods because I'm an ostracod geek. And here's just a log plot of abundance of these going back in time for about the last 150,000 years or so, 200,000 years. And what you see is that in the upper 75,000 years, there are no ostracods. So zero is basically over here, and then super abundant over to the left. And over here on the middle panel, the pink ones are this species down on the bottom here. This is a guy that likes kind of skanky alkaline lakes, the kind of thing you'd find in parts of the Great Basin today. And this one up on the top likes a little deeper water, a little fresher water. So if you look at this combined record, you can see no ostracods last 70,000 years or so, and then they become quite abundant with mostly deep water species until you get back to about 100,000 years ago, and then these shallow water mud puddle types, alkaline lake or pond types become quite abundant. So this is giving us evidence that ostracods are bottom-dwelling benthic organisms. They don't swim in the water column very far above the bottom, and so the lake has been stratified this whole time. But prior to about 80,000 years ago, we see these big events that I referred to earlier as mega droughts, when the lake dropped by hundreds of meters and caused it to become quite shallow at times, allowing these benthic organisms to live on the lake floor. I'm going to just show you one other data set here that is similar to this. This is from lake flies, which are, there's a fossil one here on the bottom. Normally we just find their these mandibles, these things that look like fingers here, which they're predators, and they swim around in the lake. They're very slow moving and from a fish's point of view, very juicy. And normally in the daytime, they go down into deep water. But at night they surface as part of the zooplankton. And when they mature, the larvae become adults and they fly off the lake in fantastic numbers. You'd see these things out here that look like fires. And this is actually an emergence or hatch of these by the probably billions. What you can see here in the remains of these is that they're quite abundant today as fossils, but that's because they have this refuge from fish by swimming down into the anoxic parts of the lake today. But when the lake got shallow, and especially when it disappeared or became very shallow, these organisms, which in North America, they're only found in lakes that have no fish in them, basically, they just disappear from the record. And so they're also giving us a story about how ecological change has occurred here. I'm gonna skip over, Tumu will probably be unhappy, but I'm gonna skip over the diatoms. And in the interest of time, just mention one other thing about the paleoecology of Lake Malawi, and that is that when we look at these changes, we are in a rift lake. And so people always ask, well, how do you know that the changes in water level, what's driving them? Is it climate or tectonics? This is a really fundamental question that we need to answer. And the punchline, of course, is that it's both. But by looking at a combination of what's going on in the lake with what's going on in the watershed, we can start to tease these things apart. And I'll just draw your attention. This is from Sarah Ivory's pollen work. Without worrying about what's what here, Podocarpus is a pine relative, a gymnosperm, and Poaceae is grasses. The mega droughts, which are shown by these, this is the lake level curve and paleoecological record from the lake organisms here up on top. So every time we've got one of these huge dips in lake level, we've got a big increase in grass and a big dip or decline in the amount of trees. And that's telling us that what's driving these changes largely is climate-driven desiccation and not simply some kind of tectonic process. But I want you to keep that thought in mind because it's not true for the entire record. Now here, I'm showing you that same curve I was just looking at a minute ago. And so going back, in this case, all the way to the bottom of the Malawi core, which is not the age of the lake. It's only as far down as we were able to drill, about 1.2 or 1.3 million years ago. And you can see that if you think of this as a lake level curve with deeper on the right and shallower, these mega droughts on the left, you can see that there have been times when the lake has dropped very, very fast, probably on the order of a meter per year for several centuries. You know, wrap your head around that. And also equally risen as fast at other times. But there's definitely a rhythm to this. And so we spent some time exploring the change in pattern, particularly what you see here just visually going from this early part of the lake's history until about 800,000 years ago, and then looking and comparing that with the rhythm after that period of time. And you can see that the fluctuations span longer periods of time in the more recent history. So we became quite interested in what that's all about. The other thing that we took away from the ecology is that the lake was cycling back and forth between what I'm just going to call a blue lake condition. And here's a photograph of Malawi today to give you the visual image versus a green lake condition. This is Lake Turkana up here in the upper left, and it's called the Jade Sea for a good reason. So the lake was cycling back and forth, and I've colored them here as blue lake and green lake for those of you who are paleolimnologically inclined in my seminar. We could also call these oligotrophic and eutrophic, but more complicated than that, so I won't go there. Let's just call it a blue lake and a green lake. One that has a lot of mixing and a lot of algae in the water column, making it green, and one where most of the primary production, like on a coral reef, interestingly enough, is benthic and occurring on rocks and attracting organisms to those rocks. So I mentioned this already, that the lake was in a very different state prior to about 700,000 years ago. And when we started to dig into this in more detail, we found out that the communities of organisms were telling us that not only was this lake bouncing up and down, but the way it was bouncing up and down was very different prior to this period, the most recent 700,000 years. During this period of time, every time, the last 700,000 years, every time the lake gets shallow, we see these mud puddle kind of alkaline skanky things like this limnocythery coming in during these mega drought episodes. But they become quite uncommon early in the lake's history. And what we find instead are these taxa here, Sclerocypris and Iliocypris, and a lot of mollusks as well. And these things are all living in shallow water, but they're living in shallow fresh water. And here's the rub. Okay, so if you think about that Lake Malawi, its outlet is, it currently has an outlet, which is maintaining it as a freshwater body. But if you drop the lake by hundreds of meters, of course, in its present day configuration, it would drop below its outlet level. So how do you keep a lake shallow, but also fresh under those conditions? And so here, I'm just going to highlight these intervals. And overflowing freshwater shallow lakes and marshes, that was where the lakes were either through flowing, they had an outlet, or they were only briefly closed, which is not possible for a shallow lake today. Then the transitional period in around 700 to 800,000 years ago. And then the condition that exists today where the lake in its shallow phases is salty and underfilled whenever it's in low stand conditions. And there are differences in deep lake anoxia persistence as well. Very rapid cycling back and forth, in this early phase, going to much longer periods of time between these events more recently. Okay, so what's explaining this kind of ecological cycling? A strictly climatic explanation doesn't cut it, because it can't provide us with an explanation for understanding how we could have shallow yet freshwater lakes in this modern rift configuration. And so we have to invoke some tectonics in this story as well. And that's where the story starts to get really more complicated, but a lot more interesting, in that what we seem to be experiencing, or the lake seems to have experienced, is a major increase in accommodation. And the only way that could have happened is for base level to rise, and for the outlet elevation of the lake to also have risen significantly. And that requires some kind of major change in the watershed configuration. So here's Lake Malawi today. Its outlet is to the south, ultimately into the Zambezi River, watershed. But let's look at it in the past. Here, and I'm just going to go back here. I want to draw your attention to this river coming into the lake here, the Rohuhu River. It's a tributary of the lake today. But when you start to look at it in detail, you find out that there's all these reverse drainages in here. And it looks like at a lower elevation during the past, or in the past, Lake Malawi actually had an outlet that was probably at a lower elevation and drained directly into the Indian Ocean to the northeast. And so we see a lot of evidence for this in the form of incised meanders along this river, in phylogenetic evidence linking some of the fish in these drainages to Lake Malawi. But here's the idea, just a series of cartoons, that early on, the lake had an outlet. So when it was shallow, it was kind of a marshy but shallow lake. And that at some point, through rift shoulder uplift, this was cut off, and the lake level rose considerably. The Rohuhu River became an inlet, and then here's our coring site at that, our deep lake coring site at that star, and here's what the lake would have looked like during these mega drought periods of time, when it would have been reduced to a tiny fraction of its present size. So this kind of cycling back and forth of extreme lake levels is it's a recurrent feature in the history of African rift lakes. This is a picture of some diatomites interspersed with paleosols in central Kenya, one of our HSPDP drill sites. And these are processional, actually. You can see three of them here. Here's a diatomite, here's another one, each of them spaced about 20,000 years apart. Very typical rhythm of lake level cycling in these lakes today. And when you look at them up close, you can see again in outcrop some of these fine deep water laminations in the deep water diatomite phases. Another couple of drill core pictures just to show you just how exquisite and how abruptly these changes can occur. So these speckly things right in here, those are pyrite crystals, but this is a very shallow water marsh deposit. So stratigraphic up is to the left. This is from Lake Turkana. And then an immediate flooding surface above that into organic rich deep water laminated muds. And seeing a similar kind of thing over here. So desiccation followed by, or big lake level fluctuations is really a theme, a constant theme of a lot of these lakes. Okay, so I mentioned this earlier, that Malawi is hyper diverse, but in different ways from its sister lake Tanganyika. So here's Malawi, here's Tanganyika. You can see the numbers in these pie charts are just through the roof. Malawi has it's probably even more than this. It's probably close to 1,000 species of fish, but it doesn't have very many endemic genera. And that's what I'm plotting here. So the divergence is very broad. There's lots of speciation going on, but it's not very deep in time. We want to understand what's going on to cause that to be so different from Tanganyika, where there's lots of genera, of endemic genera of fish, genera that are only found in this lake, but there's also endemic genera, all these black lines here in all these other things, snails and bivalves and ostracod crustaceans. So something has permitted these species to persist and continue to diverge over long periods of time in this lake and not in this one. And the difference, just to jump to the punchline, is the Tanganyika is a lot deeper, almost 1,500 meters, as opposed to 700 meters. And it experienced these big lake level falls during these climatic episodes, but it never dried out. So while this continued to be a reservoir of accumulating diversity, here the clock gets reset. This is, again, a phylogenetic tree looking and making this point again. The Tanganyika has these very, very deep originating clades going back probably 10 million years in some cases, whereas Lake Malawi and even more recently Lake Victoria, the largest but not a rift lake, have lots and lots of species, but they've evolved much more recently as a result of these lake level fluctuations. So a guy named Tom Coker, who's a cichlid biologist, proposed a model of diversification in this lake, a three-part model where he thought that initially these things diverged to adapt to specific habitats, rocky versus sand dwelling. And then they have all these crazy mouth parts to specialize on feeding on all kinds of organisms. That video I showed you before, one of the critters that was in there was something that specializes on eating scales off of other fish. So imagine that. And then most recently, a diversification of these color patterns that make these fish so popular with aquarists. And I already noted that this radiation is younger than Tanganyika, but he had this kind of three-phased model of phylogeny. And the estimates and timing of when this happened, based on the molecular phylogenetics, was that these divergences were somewhere around 800,000 years ago, around 400,000 years ago, and in the last 75,000 years. And these numbers, keep in mind, are not based on any paleolimnology or any course. This is just molecular clock estimates based on other calibrations. So a lot of this speciation, as I said, is really fundamentally around differences in colors. You see this guy here, you might find a different color morph, a different species on the next set of rocks down the coastline. So a lot of divergence that's very, very localized. So I want to take you through the Malawi record here now and show you, and I'm not going to go through all the details of this, but early in its history, during that phase when the lake was cycling up and down but was relatively shallow, there would have been some kind of orbital insulation threshold for when it could overflow, and that would happen quite frequently. And that's why we see those frequent changes and fluctuations from blue lake to green lake conditions. Somewhere around 800,000 years ago, we think that the watershed lake outlet was reconfigured, and that's when we start to see these deep cycles starting, where the threshold of insulation that was required for overflow changed considerably because the morphometry of the lake changed. And that set up the conditions for this alternation of these blue and green lake ecosystems that we've seen over the last 800,000 years. So every 400,000 years or so, it seems that this lake undergoes one of these big, large-scale cycles from mega drought to deep lake conditions. And it's during these deep lake periods of time when we think most of the diversification goes on. So now I'm just going to have some hypothetical cladograms here showing you diversity as these expanding or contracting lines. And so during this initial evolution of habitat adaptations, that's when we think some of this initial diversification in the Coker model happened. And then probably there was a coordinated extinction event. So lots of things went extinct at that time. And then some of these shallow water organisms probably also hybridized with deeper ones when the lake got shallower. And then this happened probably again. And this seems to date pretty nicely with that radiation in the feeding morphologies that we see within habitats. And then again, more extinction events as the lake started to cycle more, especially to these low alkaline phases. And then over the last 75,000 years, we've got this radiation again occurring, going from relatively small numbers to the extraordinary variety that we have today. But in the process, a lot of pruning of this evolutionary tree. And that's probably explains pretty well why we don't see the deep diversification and the deep differences that we see in Tanganyika. So to finish up here, since I'm just about out of time, I want to tell you my pitch story. This is my elevator pitch to NSF on why they should spend a heck of a lot of money to do Tanganyika, to help us do Tanganyika. And my colleagues like Jim Russell and others are pitching this on the fantastic paleoclimate record, which we're going to get here, which is not going to extend back 1 million years, but it's going to extend back almost 10 million years. But I'm really excited about this evolutionary story as well. Where did this extraordinary diversity and ecosystem complexity come from? So this is a kind of crazy phylogenetic tree that Britta Meyer in the lab of one of my colleagues, Walter Salzberger, put together. You can see many, many species here. And we want to understand, you can see the timing of this going back 10 million years. That matches very closely with our estimates from geological evidence of the age of the lake. So all of this diversification has occurred over that period of time. And we think that this lake could answer some really fundamental questions about the anatomy of one of these adaptive radiations, where we could integrate the fossil record, maybe the ancient DNA, and there's some people in our group that are working on that now, within the context of a constrained and chronologically controlled limnologic history, like I showed you for Malawi. We could understand what all these crazy shells are all about. Why do we find these very thickened snail shells that today seem to be a result of escalation, escalatory evolution? In freshwater, it's not uncommon in the old world to find, or even in the neotropics, to find freshwater crabs, but they usually have little tiny pinchers. In Tanganyika, they're like Arnold Schwarzenegger-sized pinchers, and they're used to crush these snails, much like what we see in the ocean. So again, this marine convergence. How and why and when is this lake undergoing these kinds of changes? The fossil record can tell us something about that. And we can learn something about the origin of these deep water and low oxygen adaptations, and test models that I didn't have time to get into about this repeated species pumps, why species diversification expands and contracts over time. And again, potential for DNA applications. And probably goes without saying that East Africa is where humans evolved, and we're going to have a lot to say about that, I'm sure, in terms of environmental context. So here's our plan, and I'm going to stop after this. If anyone wants to see more slides, I can show them, but this is what we want to do with this, this new project. We want to drill, instead of a 400-meter hole, we want to drill a hole that goes down about 1 kilometer or a little more. And this lower part, this more transparent part of sequence 1 is the late Miocene portion of the sequence. This portion up here, sequence 2, 3, 4, 3 through 5, these are Pleistocene cycles in the lake, we believe. And this is the Pleistocene with those big fluctuations in lake level history, but no desiccation. And that's really important for the story. So we want to expand our record of East African evolutionary biology, but also, of course, paleoclimatology going back some 10 million years. And I think with that, I'm going to skip over these last couple of slides. I will just actually, bear with me, return to one point here, and that is this point I made at the beginning about how this really is important. Having this deep time perspective is important for understanding where we're going. This is data from Jess Tierney's Tex 86 record from Tanganyika over the last 150 years in this particular image, going back further in other studies. And another text study, warmer is to the bottom here. But what I'm showing with these other curves here is, that's declining here, is a proportion of maximum value of the lake that is oxygenated. So if you look over here, here's some of these weirdo deep water snails. And here's where they were living in 1900. Here's where they were in 1960. And here's where they were in 2000. And you can see that the deepest depths where things could live is getting progressively shallower and shallower over time. That's a consequence of warming, not only affecting productivity, but also preventing oxygen from cycling down into deep water and destroying a large part of this ecosystem. So we're seeing this today in real time, and going back into deep time is going to allow us to understand the dynamics of this under different climate states, which sometimes are analogous to what we're headed for in the future. So with that, I think I'm going to stop, and I won't go through the summary terms, but let me just skip over and say thank you very much for your attention, and I'm happy to take any questions.
Woo! Yay!
Happy Earth Day. And shout out to Joellen for her editorial today, since I hear her there.
Thanks, Andy. And as usual, you can either raise your hand and use that function to ask your question to Andy directly, or you can type it in the chat and I will relate the question to Andy. Maybe some students can go first, if there are any student questions. Okay, maybe we'll get to the student questions. I'm going to ask you a question, Andy. I'll start off.
Sure.
That was a great talk. I'm curious if, and I know your drilling project is probably gonna, it's part of the pitch towards why you want to drill. But I'm curious if you have any type of insight into the first phyla that colonized these lakes. Like, do you know what type of phyla initially might have diversified and then led to the development or the burgeoning of an ecosystem? Are there ways to even get at that?
Yeah, it's a really interesting question. And there is actually a project that I'm kind of peripherally involved with. It's a Swiss-led project that Ole Seehausen, who's a cichlid biologist, and his postdoc Mo Muchik are leading. I have a small bit in it to work on the ostracods, although we haven't found any so far. So I haven't had a whole lot to do in this project, but they're working at Lake Victoria and they're asking this question. As I said, Victoria has this huge species flock of hundreds of species, but it only dates back to about 15,000 years ago. And before that, the lake was dry. So all that diversity, probably some 400 species of cichlids have evolved in the last 15,000 years. And sadly, a lot of them have gone extinct because of the introduction of the Nile perch predator. But so they wanted to know, and they were trying to ask exactly this question: what is the incumbency? What came in first? What was the order of invasion? Now, you got to keep in mind that this is not a petri dish that's sterile that you're starting with. You're bringing in organisms from the surrounding watersheds that are quite adaptable. And in fact, if you go, one of the really cool things about Tanganyika is if you go up into a little stream say, a stream coming into the lake right here, and you collect the invertebrates or fish, you will find almost only cosmopolitan species of things that are widely distributed around Africa. And so, those are the things that are getting into the lake. These things that are living in the lake that are endemic to the lake today are largely restricted to it. Only in a few circumstances are they getting into headwaters. They do go into the outflow, interestingly enough, but it's almost like people have talked about this, almost like there's a barrier or a fence here between what's living in the lake and what's living here. So initially, the colonizers would have been a variety of different phyla, from arthropods and mollusks and fish, vertebrates, but they would have been the generalist riverine or shallow lake and marsh species that would have occurred in the region at that time.
Great, thank you.
And interestingly, you see exactly that same pattern around Lake Baikal, which is pretty cool.
Yeah, no, I've always been taken by the hypotheses of birds catching fish and then dropping them inside these lakes, and then that's how you introduce a non-cosmopolitan colonizer. But anyway, so you've got a couple of questions in your chat, Andy. So Tim asks, do the inlet river ecosystems act as refugia and sources of basal fauna over the extinction events?
Yeah, almost certainly they do, and that we see that in the Victoria. You might have noticed it's called the Victoria superflock, because there actually are connections between the cichlids in Victoria and some of the surrounding wetland, marshland areas, and even into the northern western rift lakes of Albert and Edward. So yes, we think that the upland rivers are the refugia during the drying out of the Victoria and a lot of the other lakes that occurred. That's not part of that Malawi mega drought story, but it's an LGM LGM time period, I should say. It's not necessarily driven by that, but around 20,000 years ago, these lakes, a lot of these lakes are at relative low stands, and then they start to rise around 15,000 going into the African human period. So before that, where, who was hanging out where? Probably in upland areas that were a little wetter, that could sustain some permanent water flow. And then those were certainly the colonists that were getting back into the lake. Malawi didn't dry up during that 20,000 year ago period, but it dropped by probably on the order of 200 meters or so. Same with Tanganyika. When it went almost completely dry, when it became maybe 50 or 100 meters deep and alkaline, it would have had to been recolonized by the surrounding uplands.
So Andy, Joellen has a question, and she's challenging you to a beer bet. Well, you can ask your question yourself.
I can, yeah. So what is happening? It's really interesting, and you can see there's a really nice USDA website called PCAD that has lake level fluctuations measured by JSON, TOPEX, and other satellites, and it goes back now, I think, about 30 years. So it's a beautiful resource if you're interested. Tanganyika has been going up in recent years, but that's not true of all of these lakes. So it's hard to, and as Joellen is probably very aware, Sharon Nicholson and others have tried to make sense out of the patterns of and deconvolving the patterns of lake level fluctuations. And it's complicated, I will say that. But Tanganyika has been going up. Tanganyika has an outlet, and it's been relative to these huge fluctuations, relatively stable since the 1870s. That is to say, it's been open for that entire time. Its outlet is currently about 4 or 5 meters deep, and it is it was partially controlled by a barrage that was built there in the 40s that's falling apart. And so the dynamics of this are complicated by sort of some human meddling, but generally speaking, you can't explain that whole lake level rise of several meters just on the basis of that.
So what do you think, though? I'm just, the amount of effort and time you spent and the paleo records, et cetera. I'm curious what you think, what's your bet? What is your beer bet for, does this continue to go up? Are we looking at flooding, et cetera? No, really.
Yeah.
And no, seriously.
Yeah.
What's your guess here? I'm looking at the climate indicators. I'm looking at the climate modeling. We generally are seeing increased sea surface temperatures off Africa and something that's starting to look like a warm pool, which is not predicted by most of our climate models. You're seeing suppression, though, in the monsoon, a delay in that monsoon wind reversing the currents off, Oman and Somalia. So I gotta say, I don't have a good beer bet for.
No, I don't either. And based on the last 30 years, you can pick your lake, and I'd encourage you to go to that Peakhead site and look around. There are about, I don't know, 10 of the lakes that are there that have good data going back about that period of time, and it's really hard to see a consistent pattern. That Tanganyika, it's hard for it to go up a lot, for one thing. I mean, because it does have an outlet, and like our North American Great Lakes, once a lake has an outlet, if you raise the level, it's like a check valve. More water is going to spill out of it. It's very steep sided in most places, so I'm not too worried about extensive flooding, but it could go up a little. It could certainly, go up by meters. And what happened, and this is kind of instructive, in that during the Little Ice Age, when Tanganyika was down maybe 25 to 50 meters below where it is today, and the outlet was closed off, the outlet channel actually became completely clogged with debris, and it became ombrotrophic, like a bog. So the plant leaf litter and plants and everything else that was in there built up to a considerable level. And when the European explorers started coming to Tanganyika in the 1870s, there were conflicting reports whether the Rohuhu River, which is the outlet today, was an inflow or an outflow. And there was good reason why there was confusion about it, because it was probably actually flowing into the lake at the time. The lake was continuing to rise during the early to mid 19th century. And when it reached the level of all that debris that had built up over the centuries of the Little Ice Age, it very quickly flushed all that stuff out and caused the lake level to drop within a year or two by about 10 meters. And it sat at about the same level with these little fluctuations since then. So you do see climate climate-driven changes that are ENSO related, for sure. 1998, the lake went up by about 1.5 meters. But there are also these really interesting things that are harder to explain. They're not ENSO driven, like the 1961 event, when almost all of the African Great Lakes were quite high, and there was some flooding at that time. So let's see, George, your video.
Let me just read out the question for the sake. So George Davis says, great talk. In your video of abundant fish, there were abundant big blocks and chunks of quote unquote rock. Where did they come from? Any mass wasting along steep boundaries?
Yes, absolutely. And if you just look at this picture here, you can get a sense of the relief. We always say in limnology, if you want to know what the bottom's like, just look at the land around the lake, and it's probably down to the mud line, a similar angle. So you can be in a kilometer of water in Tanganyika, a kilometer offshore. It's very, very steep sided, and in a lot of places it's just vertical cliffs. And you do have, as a result, a lot of mass wasting. Sometimes that stuff gets triggered. 98, we had a tremendous amount of landsliding, and that was exacerbated in the densely populated areas around the lake because of deforestation and watersheds and people, farming on steep slopes. But yes, you get a lot of rockfall and landslides, as well as big gravity flows, big debris, underwater debris flows just offshore from those kind of places.
Okay, we're about 7 minutes over, but maybe we have time for one more question if anyone has it. If not, for the students over here, I would highly encourage you to go and accost Andy in his office or send him an email in the time of COVID. And basically you can ask your questions directly to Andy. So thank you very much, Andy. That was great.
Thank you.
Really exciting. Way to go. Woo! More.